Let's have now the antigen antibody interactions. Okay, antigen antibody interactions. Now, antigen antibody, they interact up in three zones. One particular zone is termed as like this, and like this. So this particular one in the center is said to be a zone of equivalence. There is a zone which is termed as prozone, and there is a zone which is termed as postzone. Prozone, you know from the previous knowledge, it is termed as antibody excess state, and postzone is termed as antigen excess state. Okay, so this is antibody excess and this is antigen excess, that is a postzone. The real action, the real reaction or action happens in which zone? Zone of equivalence. Why is it so? Because in this particular zone there is a formation of proper lattice. There is a formation of what, guys? Proper lattice. Proper lattice is being formed up in which particular zone? The zone of equivalence. Okay, now the reaction happens in postzone also, but they are not forming a proper lattice. Then how to make them have a proper lattice? So guys, to bring the prozone into this zone and to bring the postzone into this zone, what you need to do? You need to do the dilution. Okay, and when you do the dilution, the prozone antibody axis will come to this particular state, and according to that you can put the antigens. So there will be a proper lattice formation. The same thing goes for the postzone also. Postzone is antigen axis, and this also should be brought up in a zone of equivalence. Then only the proper lattice is produced. So what is this hypothesis termed as? This is termed as lattice hypothesis. You call this as what, guys? You call this as lattice hypothesis. Fine. Now, there are two words here. One is termed as affinity and one is termed as avidity. Okay, what is affinity and what is avidity? Affinity is the intensity of attraction. What is affinity? It is termed as intensity of attraction between antigen and antibody. Okay, affinity is the intensity of attraction between antigen and antibody, whereas the avidity is the strength of the bond. The strength of the bond after this complex formation. Which complex formation? Antigen antibody complex formation. Okay, so you got this particular point, that what is affinity and what is avidity? The affinity is intensity of attraction between the antigen and antibody, and avidity is strength of the bond between antigen antibody complex formation. Okay, means to say what? Affinity is sort of a love story, and avidity is the marriage. Means in the love, what you have? Attraction, intensity of attraction, and avidity is marriage. So there is a strength of that particular bond, antigen antibody complex formation. Is it fine? So this is what is termed as affinity and avidity. The first one which we'll talk here is precipitation. Precipitation reactions. We'll talk first of all regarding which particular reactions, guys? Precipitation reactions. In the precipitation reaction, there is a soluble antigen with mixes with antibody to produce insoluble precipitate. What it produce? Insoluble precipitate. The soluble antigen mixes with antibody to produce what? Insoluble precipitate. This is what is observed in the precipitation reaction. Okay. Now, this is important. Then which sort of antigen is there? Soluble antigen. What is the typical example of a precipitation reaction? That is Escali Thermo Precipitation Test. And this Escali Thermo Precipitation Test is done for Anthrax antigen. This is being done for what? Anthrax antigen. Okay. Besides this, you have certain modifications of precipitation, and the various modifications of precipitations, one of them important one is flocculation. And this particular flocculation is what? This flocculation is the modification of the precipitation, and a typical example of flocculation is VDRL. And what is VDRL? VDRL is Venereal Disease Research Lab. This is the test, and it is a slide flocculation test. It is a slide flocculation test. Okay, it is a slide flocculation test. And obviously for this slide flocculation test, we need a microscope. And under that microscope, what you can observe? The floccules. Is it fine? So this you need to know. Other than that, what else? Other than that, in this particular one, you need to remember this, that VDRL CSF, this particular one, VDRL CSF, this is there, which is termed as investigation of choice for neurosyphilis. It is termed as investigation of choice for neurosyphilis. This we already have done in the bacteriology. Other than this, regarding VDRL, you can remember one more thing, that this is the best test to assess prognosis of syphilis. Okay, this is the best test to assess prognosis of syphilis, means the treatment prognosis, that how much it have gone after the treatment. That is best assessed with the help of VDRL test and looking out for its titers. Is it fine, guys? So this is regarding the VDRL. This is a modification of the precipitation, and this particular test is termed as what? Flocculation. This particular test is termed as what? This particular test is termed as flocculation. Okay, guys. Now, certain other modifications of precipitation. One is termed as this way, that you have a agar gel. Okay, what you have? You have a agar gel, and in this particular agar gel, you have made two holes like this. Okay, in one of the holes you put up the antigen, and in another one you put up the antibody. Fine. In one you put the antigen, in one you put the antibody, and you allow on this particular agar gel to move them towards each other. Okay, suppose this is, I denoted by this color, so it moves towards this, it moves towards this. Let's denote it by a red one. Okay, so when they meet up with each other like this, they will produce what? Lines of precipitin. You call them as which particular lines, guys? The lines of precipitin. And these particular lines of precipitin, they takes hours together to come, because this particular precipitation reaction is slow. But what you do, you just run down the electricity down to this particular antigen and down to this particular antibody. Electricity you run under them, and what you observe after the running of this electricity, that the reaction which was happening up, the formation of lines of precipitin in hours, that particular reaction happens in minutes. The reaction happens in minute. So basically, you should know this, that after this electricity being run down, this particular interaction, it becomes fast, and that is what is the basis of CIEP. That is what is the basis of which particular reaction? CIEP. And what is CIEP? Counter Immuno Electrophoresis. It is termed as Counter Immuno Electrophoresis, also known as CIEP. Means if we run down the electricity in the antigen antibody, the reaction fastens up. And that is what is the basis of this CIEP. This is what is the basis of this particular reaction, CIEP. Okay, guys. After this, we'll talk about the Rocket Electrophoresis, which also is based on this precipitation stuff. Rocket Electrophoresis, or you can say phoresis also. So this particular one, electrophoresis, phoresis, they are the same stuff. Some write E, some write O, so you can write up either way. So electrophoresis, rocket electrophoresis, phoresis. This particular one, how we perform that, first of all, you see that in this we take the same, your graph structure. In this side, x-axis, this side, y-axis, okay. On this particular side, what you have done, you have taken the agar gel. Okay, you have taken the agar gel here, and in that agar gel, you have made some holes like this. Okay, you have made some holes, and this side of the agar gel, this is all agar gel. In this agar gel, what you have put with antibodies, you have put that. Okay, so basically in this particular full side, what is present? In this full side, there are antibodies in the agar gel. Is it fine? Now, in this particular holes, what you put up? Antigen. Suppose in the first one you put 5, then 10 units, then 20 units, then 40, then 80. Okay, so this basically are what? They are units of antigen. What you call them as? You call them as units of antigen, and they are being pushed and they are being put in the ascending order. Means for the first one 5, then 10, then 20, then 40, then 80. Yes or no? Now, when you do that, when you run down this particular one, electricity now, after putting up this one, you run down electricity because electrophoresis is there or phoresis is there. Now, when you run down the electricity in this, what you observe? You observe formation of rockets like this. Okay. And in this, what you observe? That if the 5 units of antigen are there in this particular one, the rocket size will be less. When the 10 units of antigen is there, the rocket size is bigger than that, 20 bigger than that, 40 more than that, 80 more than that. So this particular thing is termed as what? That when the 5 units of antigen is there, it will bind to the 5 units of antibody from this particular pool. Okay. 10 units of antigen binds to the 10 units of antibody from the pool. 20 units of antibody from the pool. Like 40, like 80, all that in this way they combine up and they produce these particular rockets. So this particular rocket electrophoresis proves up which theory? This particular one proves up the lattice hypothesis. And what is that lattice hypothesis? The zone of equivalence. Okay, what is that? Means whenever the antigen and antibody, they are in equal concentration, then only the proper lattice is produced. This is what is observed in this lattice hypothesis. Okay, this is what is observed in this lattice hypothesis. Okay, that's what I already told up, and this is being thoroughly proved in this. Why? 5 units, then only the size is this much. 10 units from this pool, so size increased up. 20 more, 40 more than that, 80 more than that. This is what is termed as rocket electrophoresis, and these rockets are formed up by which particular basic principle? The precipitation. Okay. Now in the precipitation only, certain immunodiffusions they also come. Okay. In the precipitation only, certain immunodiffusions they also come, and the first one is single diffusion in one dimension. Single diffusion in one dimension, and the name of this is termed as what? Auden. What do you call this as? Auden procedure. Now let's understand that what is it? Single diffusion in one dimension. So guys, here it is a test tube. In this, there is antibody, and this antibody is fixed in what? In the agar gel. Over that, you put up antigen. Okay, over that, you put up antigen onto this antibody in the agar gel. And what you observe, that when they meet up, there is a formation of lines of precipitin. Okay, so there is a formation of what? Precipitin line or precipitin. Is it fine? Now, why it is termed as single diffusion and one dimension? Single diffusion means only one thing is moving, only antigen is diffusing. One dimension means direction is this, the direction is this, and this is termed as single diffusion in one dimension. This is first thing. The second thing is double diffusion. Okay, you got my point. Double diffusion means antigen will also move, antibody will also move, but the direction is same. That is one dimension. Okay, how to go for it? Go like this. That again, test tube is there, in which in the center you have put up a agar gel, agar slab also you can say. Okay, here it is the antibody which is free this time. Here it is the antigen over it, and in between they meet up into this agar gel, and they produce the precipitin band. In between they move and produce what? Precipitin band. And what is the logic of it? The logic of this is both are moving. Antigen is also moving, antibody is also moving, but the direction is this y-axis only. Remember, okay? So that's why we say double diffusion in one dimension. This is termed as what? Double diffusion in one dimension. Now coming to the third one. The third one is what? In the third one, you have a radial stuff like this, that in this particular pool, you have antibody in the agar gel, and like this you produced up these particular what? Cuts into that agar gel previously only. In them what you put up? Antigen. Okay, here also antigen. Everywhere antigen. I am represented by A. Okay, so everywhere you put the antigen, the antibody is fixed. Where? In the agar gel. So means what? Only one person is moving. Antigen or antibody, what person is moving? Which one? Antigen, because antibody is in the agar gel. So this is also what, guys? Single diffusion. Okay, this is also what, guys? Single diffusion. But remember one thing, this is not one dimension. It can move this way, 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 it can move this way. It can move in any way. So basically, it can move 3D. But you can't see the 3D, you can see the 2D. That's why we say single diffusion in two dimensions is this particular one, and this is termed as radial immunodiffusion. It is termed as what? Radial immunodiffusion. Okay, this is termed as radial immunodiffusion. Okay, now the last one here, the fourth one is what? That double diffusion in two dimension. Okay, double diffusion in two dimension, and the typical example of this is Allegel Precipitation Test. The typical example of this particular one is what? Allegel Precipitation Test, in which you take a filter paper strip, okay, which is having the antitoxin, and on this you put up like this the three streaks. The first streak is of a positive control, positive sample. Third streak is of a negative stuff. Negative means non-pathogenic. Non-pathogenic. Positive ones pathogenic. and you want to see the patient streak also, patient sample streak also, does it producing the toxin or not? Okay, so we have taken what? A non-toxigenic, means this particular streak is of that particular conjugated diphtheria which produces toxin. This particular streak is of one which is a non-pathogenic conjugated diphtheria which is actually non-toxigenic, which is actually what, guys? Non-toxigenic. This one is toxigenic, and this one is patient sample in the center. Now what you observe, that it is coming in the contact with the antitoxin and it starts producing up certain lines, and these are termed as lines of Precipitation. It is termed as what? Lines of Precipitation. What I want to say in this, that the antigen is there in this streak, it is pathogenic, okay, and antibody is there in this particular filter paper which is having this antitoxin, and this particular antigen is moving in various directions and producing what? Lines of Precipitation. Means it is moving in different directions, means 2 dimensions, and everybody is moving, antigen also, antibodies also, so that is termed as double diffusion. So this is what is there, double diffusion in 2 dimension. And suppose this patient is having diphtheria, it will produce what? Toxin. The same story will happen with this also. But this story will not happen. These lines of Precipitation will not be produced in a particular negative control which is having this non-pathogenic Corynebacterian diphtheria, also termed as non-toxigenic one. Means it does not produce toxin. If it doesn't produce toxin, then obviously it will not be combining with antitoxin. It will not produce what? Lines of Precipitation. You got my point or not? So we do in this particular test the 3 streaks over that particular filter paper which is having the antitoxin, okay, and you observe that whether that particular strain is producing toxin or not. So suppose you have taken a positive control, toxigenic one, so obviously that will produce toxin. It will combine with antitoxin from this particular filter paper and they will produce what? Lines of Precipitation. Patient sample, suppose it is having diphtheria and we are suspecting that strongly, then that also this will come. But a negative control is why? Because for the false results, it should not happen. So negative strain is also there with us, in which this is non-toxigenic conjugated diphtheria. Does it produce this particular one? No, it doesn't produce any lines of Precipitation. So this is termed as double diffusion in 2 dimensions. Means antigen is also moving, antibody is also moving in different directions, and they are producing a what? Lines of Precipitation. Okay. So guys, the first one is the scientific name termed as Auden. The second one is termed as Oakley-Fulthoff. The second one is termed as what? Oakley-Fulthoff. And the third one is termed as radial immunodiffusion. This I already told you. And the fourth one is termed as this. The scientific name is Auchterlonie. The scientific name is what? Auchterlonie, of this double diffusion in 2 dimensions. Of this double diffusion in 2 dimensions. This much is there in the precipitation. So in total, what we have observed in this precipitation, soluble antigen mixes in antibody to produce insoluble precipitate. Not a very good reactions. Why? Because precipitate is not easy to see. Then the modification of that is flocculation. Other modifications that is being used in counter immuno electrophoresis. Rocket electrophoresis also comes under this. Besides that, we have this immunodiffusions. Single diffusion in 1 dimension means only one thing is moving in 1 direction, that is Auden. Then both things are moving. Double diffusion in 1 dimension, that is Oakley-Fulthoff. Then only again a single thing is moving. Single diffusion in 2 dimensions, that is radial ones. And the last one is Auchterlonie. Double diffusion means both are moving in different directions, that is Auchterlonie. So that is also there. Allegel Precipitation Test is an example of that. So these are there, and most of the reactions, they happen in which particular zone? Zone of equivalence. Why? Because there is a proper lattice. Okay.